Hey everyone, this is Ari Witten, and welcome back to the Energy Blueprint podcast. Today, I have an episode that I am very, very excited about, a guest I am very excited about, Dr. Ted Achacoso, who is double board certified in nutritional medicine and interventional endocrinology, and is a mitochondrial expert. His favorite topic is mitochondria, and my favorite topic is mitochondria. So today this is actually kind of a little bit of a selfish podcast because I've been wanting to talk to Dr. Ted for a long time, just for my own benefit, for getting to clear up certain questions that I have where the science, there isn't a consensus among researchers about certain things, and there's kind of arguments back and forth. And I wanted to talk to a true expert in this field who is also an expert on the clinical level in the trenches and kind of what actually works in practice. So we're going to be talking about longevity, disease prevention, decreasing chronic inflammation, stress resistance. We're going to be talking about aging itself, what's actually driving that. And let's get into it without any further ado. I'm very excited to introduce you all to Dr. Ted Achacoso. Welcome.
Thank you.
Yeah. So I would love for you to talk about, maybe first to talk a bit about your background of how you got into this, because looking at your bio, it looks like you started more in neurology and neuroradiology and pharmacology and more of like, I guess, standard conventional medicine types of things.
Yes.
And then at the age of 45, you got into anti-aging medicine, nutritional medicine. So what prompted that shift for you?
Actually, I'm trained in interventional neuroradiology, and I think they call that field now minimally invasive neurosurgery, and also in pharmacology and toxicology. And then here in Washington, D.C., I trained under the pioneer of medical informatics, and I did artificial intelligence research. And at the time, I was asked by another pioneer of socially responsible investing to be the senior science and technology advisor for his investment funds. And he asked me, he said, so Dr. Ted, where is medicine headed? And I said, I was 35 years old at the time. And I said, well, it's going into regenerative medicine, into anti-aging medicine, into real-time preventive medicine, where you don't have to think about vaccinations, etc. You can just do minor tweaks in your lifestyle, and you should be preventing diseases. So I said, it's all going into the direction away from illness and into health. And, 10 years later, I decided to put my money where my mouth was. I said, well, the technology is here. We could detect a lot of metabolites that now could indicate inflammation, indicate disturbances in your mitochondria, disturbances inside the cell. Because what has transpired in medicine in general is that we're very technology driven, right? The initial diagnostics that we had were all organ based. So therefore we took a look at organs, like liver function tests, for example. And then came the tests for the specialty cells, like the insulin that's being produced by the endocrine pancreas. You could test for fasting insulin now, right? But still you're looking at the specialized function of the cell. Then suddenly about 25 years ago, clinical metabolomics came about, and it's now just reaching the clinics and said, well, hey guys, remember that Krebs cycle that you just used to memorize in medical schools? Well, now you could measure all of those, and you could measure the cofactors that drive it. So suddenly you are moved away from illness of organs and dysfunction of the specific organs into the more general or fundamental cell that is the basis for your entire body. And illness medicine is not used to looking at the body that way. We're used to looking at it in organ parts or in discrete parts. In fact, one of the first experiences that I had when I was pioneering this whole technique was, they thought that I was going to steal their patients. So it's really a change in perspective that you have to do in order to say that all of these cells are actually foundational to your entire body. They are the ones that compose the organs. No one's taking care of them. So I went to Paris. I trained with Terry Hertog, who is the pioneer in using hormones for hormone balancing for anti-aging, and he also taught nutritional medicine. And after that, I just totally left wherever I was, and I said, this is where I want to be. This is where medicine is headed, as I predicted 10 years ago, as 45 at the time. And then I started this whole practice, like really, really putting in mitochondria, microbiota, everything that has been neglected in medical school, like epigenetics, chronobiology, evolutionary medicine, all of those stuff that are not taken in medical schools. I said, this is medicine 2.0. This is where we should be focusing in now. So we can actually prevent all of these diseases that are coming on, and not in a vaccination sort of way, but in a lifestyle sort of way. Like I have many patients who are very young, 30, years old, who says, I don't want to have Alzheimer's like my grandfather. What do I do? So we are able to answer these questions a little bit better now. Whereas before we just say, hey, it's a neurodegenerative disease, you can't do anything about it. But now you can. So that's my journey into this. That's why I started health optimization medicine as a clinical practice. And I really zeroed in on bioenergetics, because without energy, there's nothing that moves you. You're basically dead, right? So
yeah, and I think that's a beautiful segue into mitochondria, which is what I want to talk about for pretty much the rest of the time here.
Yeah.
So bioenergetics, for people unfamiliar with that term, what does that mean, and why are mitochondria so important in this new paradigm? And you kind of alluded to it, but there's been a shift, I think, over the last several decades away from kind of a focus just on the genes, or just on the organs, or just on the brain, and now people are looking more and more at the cellular level, and I think more and more specifically at mitochondria. So why is this shift going on towards a focus on bioenergetics and mitochondria?
Well, one of our big mistakes was assuming that the genes were going to answer everything for us. Well, because mutations of dominant genes can give you all sorts of dramatic things, right? In terms of the manifestation. But, second to the last count, we only had 26,000 genes. And the last count that I know, we had 19,500 genes. So the answer isn't really there. And looking at the new science of epigenetics, that controls the genes from outside, you see, oy vey, how suddenly we're not looking at the genes anymore. There are other factors that would affect that. But then I said, well, before anything can move inside the cell, there must be something that powers it up. So I focused on where does it derive energy from, and looking at it from an evolutionary perspective. If you take a look at the last universal common ancestor, for example, the cell wherever all other cells were derived, they actually were like encapsulations of membranes which relied on a passive gradient of hydrogen, outside, and then more or less negative inside. So there's a passive inflow of hydrogen, and then that gradient can be used to create ATP, which is a very ancient molecule. It's already being used even during the passive gradient period. And then what we did simply as animals was we actually simply automated the process. The passive gradient became an automated gradient. We started pumping out protons outside of the membrane. But it's more interesting how that happened, because initially under anaerobic conditions, or conditions without oxygen, right? You have your alpha proteolytic bacterium, which is supposed to be prototypical, the prototypical bacterium that can use glucose, for energy without using oxygen. And then there was another organism that could actually use oxygen. And the sanitized version of that is that, well, they went into a cooperative agreement, but actually before there was a fluctuating levels of oxygen in evolution, right? So there was a kleptogene inside the mitochondrial bacterium, and which aim to actually steal the ATP that's being produced from the alpha proteolytic bacterium. But then the oxygen levels rose, and therefore we have this symbiotic relationship now. But the plants are even one step there behind you. They're even one step ahead of us. They incorporated cyanobacteria, which are now the basis of the chloroplast, and they are able to photosynthesize, and therefore they don't need to move to get their energy. We need to move in order to find energy for ourselves, right? So if you take a look at what a cell really needs to do, it needs to survive, right? And for that, it needs to find an energy source, right? And then when conditions are right, it will reproduce, and it will protect itself from harm by either the primitive fight or flight syndrome. Either they attack or they actually run away. And these things are still very basic in us, inside us. But for the modern way of looking at it is like, think of it as, the amount of energy that you have, the amount of money that you have in a bank. So there is the energy that you're currently using, meaning the amount that you have withdrawn, and there's the amount in there that that's actually saved. And you should actually keep a balance between those, because you cannot be overdrawn. If you're overdrawn, then you could have conditions like, one of the topics that you mentioned, which is chronic fatigue syndrome, or otherwise called myalgic encephalitis, and now it's called something else. I think it's like
SEID. What is it? Systemic exertion intolerance disease.
Systemic exertion intolerance disease.
Yeah.
Yes, that's it.
Yeah.
So, but I kind of like the myalgic encephalitis, encephalomyelitis, because it's actually very descriptive. There's inflammation of the neurons and there's inflammation of the muscles. But, let's see what the IOM comes up with next.
Yeah.
So for bioenergetics, we see that we actually need to have an energy source somewhere in order to do all of these functions, right? And so when you see that energy being produced by the anaerobic cell, you see it's being produced very inefficiently. But there was use for that. That was used for low oxygen environments. And you could still see that being used extensively by sprinters. Sprinters, for example, have huge muscle cells, and which is filled with glycogen. They, and they could run at really, really very, very fast speeds for short periods of time because they could use the glucose as fuel, because at that particular speed, it's very hard to pump oxygen and blood into your muscles. So, and that's the advantage of that. But if you're doing endurance running, etc., then you want to have more efficient mitochondria, and fuel it up that way. As I said, we're always looking for this in order to do what we want to do, in order to do what the cell wants to do, which is first and foremost to survive. And after surviving, it wants to thrive, right? So it has to be it likes to find the right environment, or there's food, and so on and so forth. And then after that, it could reproduce. And one of the other things that you wanted to touch on was the cell danger response, right? And if you take a look, this is like two things. These are like our current parasympathetic and sympathetic nervous systems, and the response is sympathetic. Essentially, you fight or flee, right? If we just have it in an organ system, and we have it in a cellular, molecular level, at the level of individual cell. So, looking at bioenergetics from this point of view, as a fusion between two organisms, and energy production is done that way, makes you see immediately that there is a symbiotic relationship. And the thing that I don't want to get lost is that these are bacteria. So I call mitochondria endosymbionts. We have 100 quadrillion of them in the body, that's powering you up, for everything that you do. And so when you take a look at that, I really like to touch on how bacteria actually operate. They don't do cell division. They do fission. They divide like regular bacteria. And mitochondria have circular DNA, and there's about 1 to 15 per cell. It's average of 5, right? And some of the cells in the body have a high number of mitochondria, like the liver, about 1 to 2,000 per cell. The average is about 500 mitochondria per cell. And the red blood cells used to have mitochondria, but no more, because the mitochondria is actually the origin of the heme in blood cells. So you could see all of these functions of the mitochondria that are already have been transferred to them, aside from being an energy source. As an aside, and I don't like this to be forgotten, for the vitamin D freaks out there, the vitamin D is actually activated in the inner membrane of the mitochondria. Okay, so I don't like that to be forgotten, because it tends to get lost in the equation. So when you have mitochondria like this, dividing that way, so you could have mitochondria that actually will have mutated DNA on one side and healthy DNA on the other, when they divide, since they divide stochastically or randomly. And that division, can give you healthy mitochondria or defective mitochondria. Defective mitochondria are automatically actually, if your body is working properly, then they're automatically removed, right, by your garbage collector. Actually, they kill themselves by a process called Mitophagy, and they go away. But the mitochondrial DNA mutates very rapidly. So, and that can give rise to variations in even how much energy you produce. So it's like having a microchip and having variants of the microchip depending on how much speed the microchip can. They all will operate within a certain range, but certain combinations will provide you, a better throughput, and certain combinations will provide you with lesser throughput. And that is actually exemplified by what's called the mitochondrial haplotypes, right? So in energy production, the mitochondria will have different types of haplotypes, as described by Doug Wallace, in his studies, where he traced the mitochondrial Eve, since mitochondria come only from women, right? He traced it from Africa. That's a haplotype of L, and then as you move northward, you find that a measure called coupling coefficient of the mitochondria changes. So coupling coefficient is this: if I eat an equivalent amount of glucose, this is the amount of ATP that I produce. If I were a runner from Kenya, I would probably have a 1:1. I would convert it very, very quickly. However, if I lived in northern climate, which is very, very cold, then suddenly I will have to use some of my gradient for heat production. And that heat production will actually help me survive the winters in the cold climates. So suddenly you could see the efficiency of energy production, because now it's being used to produce heat. More recently, as recently as 2 years ago, you probably read this, mitochondria are actually producing light. Mm-hmm. So they postulated that it's being used. Much of my work in artificial intelligence was consciousness, and we were looking for a way where how is coherence produced, but in a much faster way. It's probably from light emitted by mitochondria, but I don't know. I'm just guessing at this point in time. So if you look at that now, you have many variants of mitochondria that are produced because of the bacteria-like action, fission. They actually exchange DNA material. So they actually exchange DNA material with the main nucleus of the cell, which is the anaerobic cell. Remember, I said that they were two organisms, right? So what has happened then is that it transferred much of the structural protein formation into the nucleus of the cell. So, and the energy, or the energy circuit, is preserved within the mitochondria, but those that create its structure, much of it has already been transported to the nuclear DNA. And the reason for that is really very simple. it's an energetic equation. It's so much, it requires so much energy to produce structural proteins. So if you're going to produce rapidly adapting mitochondria, depending on energy conditions, you just replicate the circuit, and allow the nuclear DNA to produce the encapsulations for you, or the body that the circuit is going to inhabit, so to speak. So, and then another is because they're bacteria, they move, right? And so in these types of movements, it's necessary. For example, your neurons are very long, right? And they have to traverse this entire length in order to get into your synapses, for example, to power up the release of those neurotransmitters. And then, as I alluded to earlier, they have a way of killing themselves if they're no longer useful. And it's actually interesting because I always like to remember their trigger is really nothing more than a loss of that proton gradient, a loss of electrical charge. You could see the loss of electrical charge, and when the body sees that it's too low, then it douses itself with the proper molecules to say, okay, I'm marked for killing myself. Mm-hmm. So, this is a pink Parkin type of things that you probably have been reading about. So because of this, fission, fusion, they fuse, they form rings around the nucleus to power up all the cell functions. They move, right? In order to power up all parts of the body. And then they kill themselves. So these are typical bacteria actions. And if there's anything wrong with any one of them, then you develop diseases, right? If there is no fusion, you have a very, very small mitochondria that are not able to generate enough energy. If there's no fusion, then you cannot form the large bridges of mitochondria that are needed to actually power up, huge sections of the nucleus and the large parts of the cytosol. Yeah. If there is no movement, then you can accumulate, garbage on one side and and nothing on the other, and your cell is going to die at that particular point in time.
Yeah. Well, this is a beautiful just tour of like millions of years of evolution compacted into 10 minutes. Yes. But I also know that there's probably a significant segment of viewers who we're only able to understand half of what you just said because it's just too advanced for them. So to make this really relevant to people, how does that all translate on a very practical level? Why is it to somebody listening to this who they're having symptoms, they have chronic fatigue, they are having various health problems? Why is all of that, everything that you just explained and what mitochondria are and what they do, how is it relevant to them on a practical level in terms of their health?
That is relevant on the other side of what the bacteria do, which is their function, right? What they do for you. And they do really just a few major things. One is the production of energy, which is what it's known for. And as I said, I don't like looking at the mitochondria as batteries for the cell. I like looking at them as kitchens that cook the food for you. Mitochondria are the only organelles in your body that can actually metabolize your fat. So these are the things that it does for energy production. But actually one of the major things that it does inside the cell for you is what's called I don't know whether or not this is too much. It's called redox balance. It's a reduction oxidation balance. And because it uses oxygen as the final acceptor for the electron when it generates energy, any factory that is going to be generating energy is going to produce some garbage. And that garbage is in the form of rust. So that, however, a low rusting level, which is called reactive oxygen species is the name of the rust, a low rusting level, of course, nature is quite very penurious in its use of signaling molecules, right? So it will use the reactive oxygen species to actually signify to the nucleus, this is how much energy production that we have. It will use other things like ratio of your ATP to ADP and AMP, your NADH ratios, and so on. But essentially, those low levels of reactive oxygen species will signify to your nucleus how much, this is how much output, that clean output that we're having. And then if it's too high, then it's like, oh, we're already having dirty output, and the nucleus can signal biogenesis, meaning let's generate new kitchens for you because your kitchens are all overworked.
So this is already, I think, an important point that I want to emphasize, which is a lot of the way that conventional cell biology was taught for a long time was like the nucleus is the master. That's, the brain, this is the place that's dictating everything that goes on in the cell, because that's where our genes are stored. And what you're saying is actually mitochondria are doing things, mitochondria are sensing things, and then communicating back to the nucleus, which in turn influences things like which genes are being expressed and so on.
Yeah, that's why I said that you should always take a look at them as two separate organisms. Mm-hmm. They are communicating. It just so happens that one of them lives inside the cell. It doesn't mean that that it has lost its full autonomy from that cell. Mm-hmm. So a lot of people, researchers describe it as semi-autonomous, but for the most part, those mitochondrial bacteria are pretty dang autonomous, except for the fact that, they cannot build their own house. They will actually dictate a lot of stuff to the nucleus. It just so happens that our initial focus historically, again, because of technology, our initial focus historically has been on the nucleus and the cytoplasm. And because the mitochondria as an organism is very small, we tend to have that point of view. However, if you erase that and just say, well, they're equal organisms, they're talking to each other, then suddenly you will have a different point of view in looking at this too. They are in a symbiotic relationship, but not 100% symbiotic. The anaerobic cell will protect itself when it needs to, and the mitochondria will protect itself when it needs to, from the anaerobic cell. So, and this, as you can see, this is actually can be generalized as a whole. That directive of the cell, if the cell doesn't want to cooperate, it transforms into a cancer cell, right? Because it wants to survive and be independent of everyone else. So you could see all of these evolutionary drives that are in there that we have sort of masked with so many layers of things. But essentially, we as organisms are doing exactly what those organisms are doing. Oh, an important point, Ari, is this. I failed to mention at the outset that I actually subscribe to the superorganism theory of the human being.
Can you explain what that?
We are actually, yes, comprised of all of these different organisms that got into relationships with each other, and here we are forming a cell. And I think the fancy name right now is a holobiont theory. Mm-hmm. So. And that's why it's, for me, it's the only tenable way to look at mitochondria, to look at the nucleus cytosol, and to look at gut microbiota, because all of these bacteria are talking to each other.
Yeah,
and that comes us. So
mitochondria are doing a few key roles that are influencing our functions. One is obviously energy production. They're also influencing metabolism of like our body fat stores, and they're involved in that process. Like you said, they're kind of kitchens in our cells. And they're also involved in various kind of signaling roles back to the nucleus of the cell that are influencing gene expression.
Yes.
Are there any other... Yes,
I'd just like to add to that actually one thing. Mitochondria is also the calcium sink of the cell. So it's like your bones. If there's any excess calcium in there, your mitochondria will try to balance it out. So, aside from energy production and ROS production and redox balance maintenance, it does, calcium balance inside a cell. So you could take a look at your mitochondria as actually taking, as a microcosm of the function of your bone in your system and the calcium metabolism for the entire cell, which is very important, really, for heart diseases, for example, because when you get a heart attack, the biggest injury would come from the reperfusion, or when blood supply comes right back into your heart, and it's deprived of oxygen, and suddenly there is oxygen coming in, that can generate a lot of reactive oxygen species and inducing further damage. So, and mitochondria is very much implicated in that. And, one of the things that you can actually do is to give the patient a lot of melatonin. That would actually stabilize the mitochondrial membrane. But, that's the clinical part of that.
Yeah. So why, for anybody that's got lost in all these little mechanisms, why is it worth talking about mitochondria? Why is it worth doing a whole podcast talking about mitochondria? Why are they so important to health, longevity, diseases? Is there evidence linking mitochondrial problems to various diseases? Can you talk a bit about that, just so people can get a sense of why mitochondria are actually important?
So the reason why it's worth talking about is just asking, where do you derive your energy, right? So, for example, it's in the clinics, it's as simple a question as, why do you breathe, right? So when you ask a question about bioenergetics, it's basically tied to that question, why do you breathe? You breathe in order to have that oxygen, and that oxygen is actually delivered by your red blood cells to your mitochondria. And because your mitochondria uses it to generate the energy for you. And so that would be a very simple way to remember how important the mitochondria are.
Yeah, that's a great analogy.
So mitochondria would be, as I said, they're kitchens, they're energy generators, and so on and so forth. And the rest of the things that happen are actually consequences of the fact that they're bacteria, and they are actually factories that produce these things, and they will be producing, garbage and other stuff, that. So there will be consequences that happen in terms of, in fact, almost all diseases, because you need to power it up, right? So let's take a look at, if you don't have any power, say, my favorite example is a city in a blackout, right? So you have absolutely no power. You have no energy in the city, etc., etc. What the first thing that you think about is something that stinks, right? Well, there's no garbage collection, and you can think of chronic neurodegenerative diseases like Alzheimer's, for example, where there's a failure of the removal of the amyloid. Of course, it's a question of whether or not it's really an amyloid problem, right? But anyway, even Aubrey de Grey, in his theories of aging, for example, would say that part of it is actually a failure of removal of the garbage, not only inside the cell, but also pericellularly, or outside, just immediately outside the cell. So you could see that. And then, of course, there's no money to pay for your police force. There would be anarchy, and therefore you can have DNA mutations, because the cell cannot repair itself, right? The DNA cannot repair itself. The mechanisms try to be in protective mode, but they can't, because there's also no power to protect yourself. So you become susceptible to infections. You have a lower immune tolerance, and so on. So you're more likely to get sick with infectious diseases, right? And so if you have no power, also, then since you have no police force, etc., aside from the regulating cell growth, there's also the regulation of the various processes inside the cell. So essentially, your cell will be in standby mode without any power. The simplest way to think about it is when your phone is in low bat mode, like absolutely, and it will turn itself off if you don't power it enough. So that's what happens to us when we don't take care of our mitochondria.
Beautiful. So I want to transition into aging, and you kind of just alluded to some of it there, but there's been lots of theories of aging, and you mentioned Aubrey de Grey. He's kind of a very well-known aging researcher today that has his own theories, but there's been a transition. There used to be Harman's free radical theory of aging that was popular for many decades, and now that's kind of mostly dead. And aging researchers don't really look at that so much. Then there's the mitochondrial free radical theory of aging, which is more focused on reactive oxygen species, free radicals produced within the mitochondria, by the mitochondria. And then there's all sorts of other, kind of theories of aging, and scientists debating about which theory is the most correct. And, some saying it's about telomeres, and others saying it's about genes, and others saying it's about mitochondria, and, all these different kind of debates going on. But what in your opinion, are the major drivers of aging, and what is the role of mitochondria in that process?
Well, first of all, I'd like to say that we don't have a real definition of aging. At one point in time, aging was going to be classified as a disease, so rather than as a natural process. And now people are saying aging could be stopped. But how do we really define aging? If you take a look at it just from the body clocks, the body uses very, very different clocks. The skin uses a different clock from your eyes. It uses, so different parts of the brain uses different clocks. So, in fact, Nobel Awards were just given, for the discovery of these various different clocks. And for example, the one of the more used measures of aging would be the length of your telomeres, right? Your telomerase activity, and you belong to a certain cohort when it's longer, and so on. So there are various ways by which you want to measure aging. So what are you going to use, right? So what category are you going to lump it in? So if you're going to take a look at the mitochondrial theory of aging, then that's one thing. You're looking at it from an energetic perspective, right? And so you have to look at the genesis of the mitochondria inside your body, what haplotype you actually inherited, whether you're coupled and uncoupled, whether or not you've moved to a different environment, whether or not you change your diet, and so on, because those are all going to influence your mitochondrial function.
And just I don't want to digress too much here, but real quick, can you just talk about what you mean by move to a different environment in the context of mitochondria? What does that mean for people listening to this?
Yes, for example, if it is now been established that actually if you move to a different environment, you can accumulate mutations very rapidly for that mitochondrial DNA mutations for that environment that will adapt you to that environment, even if you don't have the specific haplotype yet for that environment. So
on a practical level, like if somebody from Kenya moves to London, would that be an example of this?
That would be an example of that. It's not as rapid as the change in your gut microbiota. Your gut microbiota can change in 24 hours based on the food that you eat. And I'm guilty of that. I'm rarely in a single time zone for 2 weeks, but that really totally fucks up my gut, my mitochondria. But it's what I mean by that. If you take a look, for example, people in colder climates, like for example, you're from Boston. As you get older, they actually don't like the cold anymore, right? Because their DNA is mutated. Many of them fly down south to Florida. And if you take a look at if you do a test of the mitochondria when they were in Boston, they had a higher coupling coefficient than, they had a lower coupling coefficient than they were when they moved to Florida.
So their cells were designed to produce more heat.
Yeah, so that's why I had to start with those basics, because this would explain things more simply.
Yeah, so when somebody's in the wrong environment and they're accumulating mutations, are those adaptive mutations, or they're bad mutations that are harmful by having this mismatch between the haplotype and the environment you're in?
They're mostly adaptive because the body, as I said, they're bacteria. They would like to have the most optimal environment possible. So when you put them in a different environment, the first thing that the bacterium will ask is, or what we ask is, where's my energy source? What do we ask when we go? Where am I going to eat? Where am I going to stay? So these are fundamental questions that are also answered by your bacteria. So the mitochondria says, well, okay, this is this is a cold climate, I need to, we need to have more of the loosely coupled mitochondria because we have to produce more heat. We used to think that it occurred very slowly, right? But now we know that within a few years alone, they can change. As exemplified by the snowbirds, they just fly over to the south, and they enjoy the warm weather that way. So these are the kinds of changes that we see. And we were talking about mitochondrial theory of aging, right? And there is this, because mitochondria powers up your cell, there's a significant number of them that needs to be alive, with producing enough energy to actually keep your cells alive. So this is one by Douglas Wallace in his paper, where he said that at around 45 years or so, that's your tipping point, where you accumulate a lot of mutations in your mitochondrial DNA, rendering the energy production inefficient. Or you become highly heteroplasmic, in other words, as a term for that. And such that the tipping point that after that you cannot recover. So, but probably that is one when you don't do any intervention. Right?
Well, just complete that thought for a minute, because what happens then? So you accumulate a lot of mitochondrial damage, mitochondrial mutations in the cell, and then what? What is the consequence of that?
The consequence is actually the consequences of that are manifold, but the primary one, which we were talking about earlier, there's a rise in the production of the reactive species, the reactive oxygen species and reactive nitrogen species. If you have an old factory, it doesn't know how to deal with its garbage efficiently, right? Because it's rickety, it's old, its collection mechanisms are not as good as when it was a new factory. So, and its checks and balances are already very loose. So, so what happens then is that it cannot produce enough power for the cell, right? The cell tries to produce more of this mitochondria, but if there are too much of that heteroplasmic mutation, meaning most of them will produce the same circuit anyway, the same type of kitchen that is inefficient, then you will have less and less of efficient mitochondria. So then there's less and less available power for your cell. Now, in the course of that you produce a lot of these reactive species. There are two types, right? Reactive oxygen species and reactive nitrogen species. And this can is actually going to signal your nucleus, says, hey, try to produce more mitochondria, but the mitochondria produces are actually more defective ones. So then the number of healthy cells die as you know, when you get older, there's brain atrophy, there's muscle atrophy, all of these things. So you lose the cells in your body. And it's,
do you mind if I just interrupt you for a sec? So that there's the link between the loss of cells and the damaged mitochondria is basically what you're saying is, as cells accumulate more damaged or dysfunctional mitochondria, at a certain point, the whole cell gets the message, hey, we're not able to produce enough energy to keep up with cellular.
Right.
So the cell commits suicide. And then over time, when lots of cells commit suicide, you actually lose cells. And as I understand it, the mitochondrial theory of aging is basically suggesting that as kind of mitochondria get damaged, then enough of them get damaged that the cell doesn't meet these demands, then you lose cells. That loss of cells through cells committing suicide is a major driver of aging.
Right, but mainly the your first trigger will be a rise in the loss of garbage collection, efficiency of your neutralizing very high levels of your active species. I think that's still very much there, but it's more of for me, it's a secondary action of the fact that you have accumulated too much of variations in your mitochondrial DNA, such that it is producing dirty kitchens for you instead of clean ones.
Okay, so let's dig into that a bit more, because the original free radical theory of aging was basically like, free radicals are bad. They're kind of rusting and damaging your cells. And the idea, the logical extension of that was taking antioxidant pills would neutralize all these free radicals and therefore prevent the damage and extend your lifespan. We now know there's lots of evidence showing that it doesn't really work that way. And, you know, lots of supplements, antioxidant supplements have shown not to extend lifespan.
And there's also antioxidant-induced toxicity.
Yeah. And the other logical extension of this theory is that things which promote free radical production would therefore accelerate aging and accelerate your demise and shorten your lifespan. One example of that would be like exercise or other forms of hormesis, which stimulate free radical production. And we know that that is also not true. Exercise actually helps extend longevity. So the free radical theory of aging, like I said, has been mostly discarded. But what you're saying in contrast is there's this kind of other picture, which is the mitochondria themselves are producing their own free radicals.
Yeah,
and that,
they are. Yes.
That those free radicals are in some way linked with aging.
Yes, because they actually damage more of the mitochondria. They actually damage more of the, if there's a high hydrogen peroxide, for example, that's traversing into the cell's nucleus, that itself can actually trigger, if it is not, I was going to say dismutated properly, if it's not taken care of properly, then it can actually cause defects in your DNA by attacking the DNA. So then all of the sequelae of diseases due to aging, more cancers, more immune diseases, et cetera, you could see it as coming from the mitochondria itself. And I think that is the important pathway that you look at. As I said, you always have to look at it as two organisms, right? So what is the effect of, if you're looking at the effect on the somatic cell, for example, or the nucleus cell, which is like looking at the effect on the body, you should look at how this microorganism is actually communicating with your nucleus and communicating with the rest of the cytoplasm. And then you'll see there how it contributes to aging in itself. So aging is either, there's also the accumulation of DNA mutations, that cannot be repaired. There is so many of this, but I said we don't have any clock by which to base it by. So the mitochondrial theory of aging is unique because it actually takes into account the energy production, and it goes by the dictum: without energy, you're dead anyway. So, so why not look at energy first, right?
Yeah.
We're so conditioned to chronological or chronobiological types of aging. And now we're looking at it from a bioenergetic form of aging, and that becomes more analog. It's not a digital kind of thing where it's on your aging, off you're not, right? So you need to have particular ratios of healthy mitochondria to one another in order to sustain cell viability. So you have healthy ratios,
Yeah, so let's dig into that a bit more, and I think this is where it transitions. I'd like to transition to get practical for everyone listening that's like, okay, enough with the theory, tell me what I can do to slow down aging and help my mitochondria. So we know that taking antioxidant pills doesn't really work to neutralize this production of free radicals by the mitochondria.
Unless you measure the levels first and see what the person needs.
Okay.
Because if there are severe deficiencies or even subtle deficiencies, you can tweak the mitochondrial function by just giving the supplements. The thing that I learned from the clinics is that your patient or your client, if they're athletes, and they're not sick, will never take the diet that you prescribe them. So you always say, here, take these supplements and go. But the big improvement that we have now is we could measure the mitochondrial metabolites. We can measure mitochondrial cofactors. We know what things make it hum, and we know what could enter the mitochondria and what can't. So based on that, then we could measure, and then we could do the proper supplementation, and we could at least optimize the function of a mitochondria currently, those that are currently alive in your system.
Okay, so let's dig into some specifics here. So what are the specific types of things that that would actually trigger more, more of an oxidizing environment, more free radical production by mitochondria, would destabilize the membrane potential. And then, and that's one kind of question that I'd like to get into first. And then the second part of it is, what are the types of things that we can do to stabilize and protect our mitochondria and decrease their production of reactive oxygen species?
I think your listeners will hate what I'm going to say, right? If you eat too much food, if you're going to put too much food for your kitchen to cook, you're going to stress out your kitchen, right? So you're going to make it throw off lots more garbage. You're going to make it more dirty, especially when you're taking in lots of refined carbohydrates like glucose, which is processed by the anaerobic part of the cell, right? And then it goes into mitochondria's pyruvate. You need vitamin B1 to get into that, and then you dump in all of this pyruvate into the cell, and you get what's called reverse electron transport, meaning everything will go backwards. It will not generate energy for you. It will actually reverse the entire process for you. So
say that again. What triggers that? Just overeating in general?
Yes, excess of the. If you're putting in excess of the substrate, pyruvate, too much acetyl coenzyme A there. It's like, oh my God, you have lots of vitamin B1, that's transferring everything in there. You're actually the kitchen saying, we're already fucking full. Why are you even doing this?
Yeah, and I don't want to digress too much into mechanisms here, but does this relate to the concept of pseudo hypoxia and the NAD plus and NADH ratio?
Yes, it's related to pseudo hypoxia. Yeah, which is I'd like to touch on that also on the other side, where how to keep the mitochondria healthy, because since oxygen is the final acceptor for the electron that's used to generate the proton gradient for energy. The one thing that you said earlier, which I think your listeners should think about, is that we're not intended to eat continuously, right? And we're horrible as a species because we have permission to eat from the time that we wake up to the time that we sleep.
Yeah.
And so one of the things that is easy to do based on this overtaxing your kitchen with food is, why don't you shorten your feeding window, and give your mitochondria time to actually rest. When when you do that, then your mitochondria get rested, and your body says, okay, let me generate new mitochondria for you, a process called mitochondrial biogenesis, right? And let me remove the inefficient kitchens for you, and let's do this. And it does that during the fasting period.
And mitophagy, autophagy and mitophagy specifically relate to that fasting window, and like having enough break from shoving food into your body that your body and your cells and your mitochondria have this time to go into autophagy and mitophagy. Can you explain mitophagy just a little bit?
Yes, mitophagy is a process by which the mitochondria actually kill themselves. It's used in several processes in the body. The first is during fertilization. That's how the egg actually kicks out the mitochondria from the sperm. It uses the mitochondria to swim, right? And the body uses that in mitophagy. A second use is in the formation of your red blood cell. There's a mitochondria there initially, and then the mitochondria kills itself in order to have the heme for your hemoglobin. And then there's mitophagy, of course, for when the proton gradient of the mitochondria that's generated is very low and insufficient. The body identifies that and marks it immediately for recycling. So it means it's an inefficient kitchen. Let's take it out. That's mitophagy. Autophagy, however, is a different thing. It's the suicide of the cell in general. Unfortunately, the trigger for that is also the mitochondria. There is a molecule in the mitochondria directly involved in the production of energy called cytochrome C. And when that is released out of the mitochondria, that's the death knell for the cell. It's like time to kill yourself. Which means it has opened a pore. There's a pore that it opens up. It has a name, the mitochondrial membrane transport pore, and when it goes out there, the cell says, well, okay, we're marked for suicide. Here's the cyanide. And this is necessary in normal balancing of anabolic processes, or processes that make your body grow, and catabolic processes and processes that remove or break down things from your body, which is all necessary, right? Which is what we do with food. We actually break down the food in a catabolic process, and anabolic process is building our muscles. It's the same thing that the cell is doing, that the mitochondria is doing. It's doing that to itself.
Yeah. So overeating in general, overeating refined carbohydrates, and
in particular,
and eating too frequently and too many hours of the day.
Yes.
All of those factors. What other factors are going to damage our mitochondria?
See, you mentioned earlier something that is not naturally done, right? Which is chronic exercise. When you over-exercise, especially if athletes overtrain, your mitochondrial, you are actually going to induce your mitochondria to produce lots of that rusting molecule called, the garbage, the reactive oxygen species, higher than what would be required to just do a signaling process. And that will actually turn on the inflammation cascade, or what's called molecular inflammation. In fact, molecular inflammation is lovingly called inflammaging, another aging pathway, but that's more on the origin of chronic diseases type of aging, like diabetes, and all of these other diseases. That's called inflammation. But the major player in there is, of course, your reactive species, reactive oxygen, reactive nitrogen species. So when you're, that's why when I used to have fights with coaches, because I would allow their athletes to rest, and would allow them to only practice or train on 2 days a week, but the classic training of the coaches, you have to train every day. No, you don't, because you have to build your mitochondrial reserves, you have to decrease the inflammation, you have to make sure that the proper anabolic processes have occurred, for example, enough for muscle growth so that there's strength, right, for whatever the athlete is supposed to do. So that's why the whole phenomenon of high intensity interval training came out, right? It's because the easy way of remembering that is very natural, really. If you were hunting for a rabbit, you're not going to run after the rabbit continuously, right? You're going to run a little bit, high intensity, you're going to hide behind a tree, run again, and then hide behind a tree, and then run until you can club the rabbit. So, that's the way we are intended. We're not intended, for example, to have a continuous supply of food. For the most part, we had parts where in our evolution where there was famine, which is now the fate of the Pima Indians in Arizona, right? They have genes that are actually used to about 6 months of famine, but now this 24/7 diabetes is like over 90%.
Yeah, and just real quick for anybody unfamiliar with the Pima Indians, they're studied in obesity and diabetes research circles because they're among the most obese and diabetic populations. And they're also unique because there's part of the tribe is in the United States, and part of them remained in Mexico. I think they're in Arizona or Texas or maybe both. And then part on the Mexican side of the border. And then there was a period where there was a famine, and then they were rescued by government rations, U.S. government rations, where they sent all these processed foods to them. And then obesity and diabetes rates just skyrocketed. Whereas the tribe still in Mexico, same genetics, but different diet, and have very low obesity rates. But anyway, just wanted to quickly add that for anybody unfamiliar with the Pima Indians.
Yes. Well, of course, this would be very easy for your listeners. Things that stress out your mitochondria, anything that stresses you out, especially your significant other or your work. That's because it drives up several things, most prominently your cortisol, right? And your cortisol levels are essentially responsible for driving up your blood glucose levels because you have to fight or flee. And before, the only purpose of that was to either clobber a tiger or run away from it. Now someone does this to you on the road, and you would get hot under the collar and you're ready to fight, right? And that will induce the same sort of stress mechanisms. It's symbolic, but it will induce the same sort of stress mechanisms as if a tiger were about to, pounce at you. Mm-hmm. So, and since your body needs to process, all that glucose and so on, then it will have to tax your mitochondria. Again, it goes back to, hey, we need to provide an energy source for a perceived threat. And we talked beforehand, about the cell danger response, which you said, and this is actually the, part of that whole thing is that, well, we're actually wired to do this, to raise blood glucose, to be able to respond to threat and so on. And then when the threat is gone, we go back to dynamic equilibrium, right? But if there is a continuous assault or continuous stress, even, for example, phototoxicity, continuous assault of blue light, even late at night, these are stressors to your system that actually will stress out your mitochondria, because your mitochondria will have to always provide the energy for you. Not only for the fact that you're going to stay up late, you're also going to be dancing and you're going to be taking some psychotropic agents. So...
And just on that point, you mentioned earlier that melatonin is actually a stabilizer of mitochondrial membranes. Mm-hmm. And that links with what you're just talking about as far as blue light phototoxicity, because if you're every night suppressing melatonin levels, that's ultimately going to lead to, over time, that means your body has produced less melatonin that can get into those mitochondrial membranes and stabilize it. So it's making your mitochondria, I think it's not just damaging them directly, but it's also making them more susceptible to damage from other stressors. Yes,
because you're going to be inducing the production of, since you're always under threat, the body, what will you do if you're always under threat? You will develop mechanisms to detect the threat earlier, right? Earlier and earlier. Look at what we're doing with our airports. It's ridiculous now. There's one shoe bomber and everyone has to take off his shoes. So the mitochondria is the same way. It will change its circuitry so that it's able to detect all of this earlier and earlier for you. Mm. Because as I said, it's an organism. It will try to protect itself.
Yeah. So what other factors, is there a role for things like poor gut health or bad gut microbiota, dysbiosis, toxins, heavy metals? Yes. Anything worth mentioning here?
Yes, actually, the whole gamut of lifestyle, right? So if you take a look at, for example, let's just take, for example, environmental toxins, right? If your liver is actually always getting poisoned by the blue dye that is in your cotton candy, right? Aside from the fact that you're eating cotton candy, I'm gonna kill you for doing that, but your liver also needs to detoxify those. So in its detoxification, it will need a lot of energy to do that. So you're actually going to be taxing not only your liver but your entire body because it has to handle the toxin for you.
On that note, I remember seeing maybe a year ago or 2 years ago, I saw a study that was looking at a food coloring dye, commonly in a lot of processed foods, showing it directly destabilizes mitochondrial membranes. So you're asking your liver to metabolize this toxin, but it's at the same time also acting to directly damage and destabilize mitochondria.
Yes, there was actually, historically, there was a drug that was introduced to the United States, 2,3-dinitrophenol. It's being sold in the black market, that can
Bodybuilding.
cause, yeah, it's called an ionophore. So what it does is it pokes a hole in your mitochondrial membrane directly. It pokes a hole in the mitochondrial membrane directly and collapses the proton gradient that was available to produce ATP. So everything is produced as heat. So when everything is produced as heat, then the person actually loses a lot of body fat, right? Because you're heating up. But it also causes cataracts very rapidly. So, and corollary to that, that's an extreme example. Very simple drugs like metformin, for example, which is a very common anti-diabetes drug, will poison complex 1 of your mitochondria, of the mitochondrial oxidative phosphorylated chain. There are various drugs that can inhibit each and every aspect of the electron transport chain, which produces the energy for you. In other words, you are fucking up your oven. So, by taking any of these drugs, for example, if you have a patient on metformin that's given by another physician, I usually ask them to stop after about 6 months, withdraw it a little bit, give the body a chance to rest, before actually going to do that all over again. So things like this are just beginning to surface in terms of how you do drug therapy. Because many of the simple things that we thought were safe are actually poisonous to the mitochondria.
And what about like statins and CoQ10?
CoQ10, for example, of course, the production of CoQ10 is mediated by hydroxymethylglutarate coenzyme A. Anyway, HMG-CoA. It's an enzyme that actually helps in the production of cholesterol. So what statins do is that it inhibits this enzyme. And in the process, you're actually inhibiting the production of coenzyme A, which you naturally produce, right? Just as an aside, coenzyme A, by the way, comes from vitamin B5 or pantothenic acid. So just in case you're wondering what the supplementation equivalent is. So if you're going to inhibit that, then your CoQ10 goes down drastically. In fact, in some patients, in 2 days, they experience muscle weakness. And for most patients, they could not identify it. They actually get low back pain, and the second day of treatment, and they go to an orthopedic surgeon. They say, well, I now have low back pain, but it's actually just due to statin administration. So, but now you can also measure levels of CoQ10. So when you're administering statins, you could measure the level of CoQ10, and supplementation is actually pretty good. There are now targeted forms of CoQ10 that can go inside the mitochondrial membrane directly.
Excellent. Any other factors worth mentioning here as far as mitochondrial dysfunction?
Yes, I could mention 7.
I'm sure that we could spend like 3 hours on this topic alone.
Yes, but I could mention 7. But on the general class, first, these are your toxins and your drugs, that occur, and then of course there's your sleep cycles. For example, if you're going to have a disrupted sleep cycle, that will actually have an impact on your energy production and your mitochondrial performance. So if you are going to push your mitochondria to produce energy for you to staying up late at night, what do you think that's going to happen? It's going to happen to them, right? You'll be actually forcing them to go on overtime. They will produce more inflammatory molecules for you. And therefore, ever notice that if you're actually, if you lack sleep, the next day you're slightly puffy, and you see that there's actually a mild inflammation that's already occurring in your body. There's the other, as we mentioned, there's continuous stress. This is in what's called evolutionary medicine, where the cell danger response is, where there's unmitigating stress. So one of the things you could get is get out of a toxic relationship, get into a divorce. I'm kidding. I'm not. So, or get a handle on how to deal with your boss, who's toxic to you. These are very practical things. When I started practice, I wasn't thinking of this, but it has a huge impact on you. Meditation practice, etc. And that has actually an impact on what's called epigenetics. These are the things, the switches inside the switches that switch on your DNA, for certain genetic expressions, for certain proteins, etc. And that's where the food that you eat, the exercise that you do, the meditation that you do, and so on, actually come in. Most physicians do not understand, for example, the epigenetic pathway of control, and so the big mistake that we did was, just jump into the genes right away without taking a look at all the other genomes or ohms that are out there. The proteome, the epigenome, the metabolome, and so on, which are more indicative of what's happening to the organism. So, the food that you eat can be very stressful. We mentioned about that. For example, if you eat biotin-rich foods or and stuff, they or broccoli and these things, they actually affect the expression of the genes themselves. So, and this affects the way the nucleus of the somatic cell communicates with your mitochondria. It says, we're all good up here. We're well defended. This is how much energy that we need. Remember, the currency is always how much energy do you need for what you need to do, and are you in distress? So this is always an exchange. Like, what's your status now? What's your status now? How much do you need? What's your status? Right? Yeah. And do we need to kill you? So in order to, this is what's called the greater good agreement, right? Oh, we agreed to be together in this, so let's all work together, right? So and that's where this whole field of epigenetics, for example, come in. Then there's, of course, the gut microbiota. If you're eating badly, if you have a leaky gut, right now there's last year there was just a paper that was published showing that the gut microbiota and your mitochondria, being both bacteria, actually talk to each other.
Yeah, I'm glad you brought this up because I was going to ask you about it.
Yeah, and that's why we know that your diet can alter your gut microbiota directly, and your gut microbiota can actually talk via short-chain fatty acids or bile acid signals to your mitochondria and influence their energy production. And for example, they can also tell them, can you please ramp up your energy production for defense of the intestinal lining? Because there is an assault that's coming in here. There's an imbalance of the microbiota. We need you to generate more energy to be able to generate new cells that are actually more protective to the intestinal lining. The body is cooperative that way, but these are bacteria that are talking to one another.
Yeah,
right? And
there are new. You alluded to this earlier, but there can be changes in the microbiome within a matter of a day or even hours. I mean, literally just eating a couple of meals of bad meals and cause a shift in the microbiome, and that's going to then those bacteria that are now proliferating are going to communicate in certain ways to the mitochondria inside of the cells in your body, in negative ways.
And when there's, for example, if the bacteria are too much in distress, and or for example they get toxic, you get a leaky gut syndrome, for example, which is mediated by microRNA. They cleave your intestinal lining. They allow what toxins called lipopolysaccharides to get through, right? And these are called endotoxins. They poison your body in an inflammatory sort of way again. So what that does is they actually, when that happens, it's a signal to the mitochondria, hey, we need to ramp up a lot of energy for repair, right? But if you're not supplying the right energy for repair, like you're giving more refined carbohydrates, for example, then more of those gut microbiota that will feed on your refined carbohydrates will proliferate. It's like, a balanced jungle, in there.
On the other
So,
hand, I've seen quite a bit of data from people who have gone long-term keto that have, done gut microbiome profiles and have ended up with pretty significant dysbiosis in that way too. I think from lack of diversity of fibers in the diet.
Yes, and that's because you're supposed to rotate this stuff. As again, you do not do chronic exercise forever and ever. You do not eat, sugar forever and ever. For example,
You don't fast forever and ever.
Forever and ever, yes.
Yeah.
And even for Ari, this is significant because I have counseled some very important people on the vegetarian diet, right? I tell them, if you want to go vegetarian, why don't you do it for 2 weeks every 3 months? Just go, because it will induce hormetic stress. It's a good stress for your body. It's kind of like for your mitochondria to be in a hypoxic environment. Like, for example, if you're training at high altitudes, right? Then what happens there is that you generate a lot of more mitochondria because they are temporarily out of oxygen. Hey, we need more kitchens to actually snag the oxygen from the air, and you develop more red blood cells too. So these are hormetic or good stressors, just like properly executed and timed exercise. So that's the body is not intended to go keto the whole day. For example, me, I do what's called a mini ketogenic diet. I'm complete within a day. I'm fasted 16 hours. My first meal is a ketogenic meal. It's absolutely no carbs. So my second meal is a high fiber, carbohydrate diet, but the net carbs are still kind of low, but it's rich in high fiber. And then I do my protein. So I step out of it every day. I step out of ketosis, nutritional ketosis every day. You can, again, the cycles for those will be long. Some will do it in a week. They replenish during the weekends and so on and so forth. But for me, I am a fan of something that you can do. Your listeners can do. It's like, yeah, why don't you just eat everything within 8 hours and be done with it if that's all that you can do. And then you'll induce mitochondrial bioenergetics. You have fresh mitochondria. You're burning fats for fuel. And the reason why you want to burn fats for fuel for the mitochondria, as I said, is mitochondria is the only organelle that can burn fats for you. And the reason for the MCT craze is that the 8-carbon chain actually can pass through the mitochondrial cell membrane without needing carnitine as a shuttling mechanism. It could just pass through and go through a process called beta oxidation, where it's shoved to produce energy. So and that's the reason why these things are currently out there in the market. Is to feed your mitochondria directly, right? So and it's doing quite a good job. I use an MCT oil powder actually every day, but that's the whole point is that you do not need to be in that state the whole time. For me, complete within a day is my mantra. I live my life that way, right? So I don't forget. So wherever I am, whichever time zone I am, okay, here's my 8 hours. This is where I'm going to eat.
Yeah. That's a great message. Thank you for mentioning that. So I want to be sensitive to time here because I know we've gone over time, and I appreciate you doing that. But can you just say a few words on maybe the link? And I know this is a big topic, so trying to condense this is a tough task. But the link between talking about kind of the role of inflammation in the aging process, the concept of inflammaging, and maybe the role of mitochondria, how mitochondrial health relates to inflammaging.
Okay.
And then actually the final thing that I'll ask you to do is just to give maybe your top 3 tips to improve mitochondrial health.
Okay. So the inflammation concept or molecular inflammation concept of aging is actually based on the production of reactive species, reactive oxygen species and reactive nitrogen species, right? So you have basically 3 main sources of the reactive oxygen species. And the first one would be your oxidases. These are enzymes that actually use oxygen. There are 2 major ones, of course, the xanthine oxidase and NAD oxidase. And then the second one is what's called uncoupled enos, or the endothelial nitric oxide synthase. And that's another pathway that uses lesser known but actually difficult to synthesize supplement, but it's already available. It uses tetrahydrobiopterin, BH4. Some of your listeners may have actually heard about that. And it's actually used to stabilize your nitric oxide synthase, which is essential. Your cardiologist knows about this. It's essential for the maintenance of your blood vessels. And then, of course, the third source would be of reactive species would be your mitochondria. And in your mitochondria, the main sources of the reactive species would be your complex 1, complex 3, complex 4, and pyruvate dehydrogenase. So those are the major sources of it. Your mitochondria has ways of dealing with this. Inside that's the role of what's called their superoxide dismutases, right? What they do is they convert this highly reactive species that can damage you into water, ultimately water, but first into hydrogen peroxide, and then by various mechanisms like using glutathione and using a catalase enzyme, and the one that's actually scary is when it combines with iron in a thing called the Fenton reaction. We don't have a way to actually neutralize hydroxyl radicals, but they occur in bursts. They occur in very sharp bursts, and they're actually used by the body in order to kill viruses. So there's a use for it, but at the same time, if there's too much of it, then you're actually going to destroy some of the circuitry inside the cell. So if you now have this and you have an overproduction of the reactive species, then they will actually cause a progressive decline. These are called the instigating agents, right? They will actually cause a progressive decline in cell function by increasing the production of inflammatory molecules. Now, that is a normal response of the body. Again, if you look at it from an evolutionary point of view, the body will try to fight the stress, with its own inflammation, as acute inflammation especially, is a natural response of the body to fight for itself, and it will. So it will bring up transcription factors like NF-kappa B, all of these wonderful transcription factors that will actually raise the inflammation in your body and so on. However, the body also knows how to balance itself. There's a pathway called NRF2, which will try to quell that whole inflammation. So there's that balance that occurs. So what happens if you are in a continuous stress, right? There's a continuous production of reactive species, is that the body's the NRF2 pathway is unable to actually keep up with the whole thing. And so, what's bad about the inflammatory cascade is that it's self-feeding. It will, the higher the NF-kappa B, the higher the response, the higher NF-kappa B will be produced. So, and that unmitigated will cause your inflammation. So then that will start attracting, of course, your classic inflammation pathway, which is like, the swelling, redness, and all of the pain and all of these other things that you would experience in chronic fatigue syndrome, in diabetes, and all of other disease conditions that are there. And they're usually the seed of many of the chronic diseases. Mm-hmm. That's what inflammation is all about. Yeah,
and just to go back to something you mentioned earlier about over-exercising and kind of chronically exercising every day, it activating that NRF2 mechanism constantly, so you never give it a break. You're constantly kind of stimulating this inflammation in the body and never giving it a chance to recover and reset.
Yes, you're not giving the NF-kappa B any rest. So you're not giving an NRF2 a chance to actually go and recover. That's why, if you're doing resistance training, two times a week would be probably enough if you're not a bodybuilder. 2 to 3 times a week, or 2 times a week separated by 2 days, and the rest will be aerobic days. Is pretty decent. If you're following the HIIT program, especially the 10-minute programs, I actually recommend at least do it at least twice a day. 10 minutes in the morning or 10 minutes at noon or some other variant.
Twice every day?
Yeah, twice. I recommend actually twice a day, because
7 days
once
a week?
although there's you've seen the concept, right, of they're considering exercise as a drug, and there's a minimum exercise dose, and they're trying to figure out what the dose is.
Right.
So, so they came up with this thing where an aggregate of like 2 minutes and 30 seconds of all out, which is on 30 seconds dispersed every 2 minutes. Yeah, that may be true for Caucasians, but what about for Asians, and what about for for those with different mitochondrial haplotypes? Then it's actually very different, right? For me, I said, well, okay, if this is a recommendation, I tend to double it. Because I think 2.5 minutes under the curve, although dispersed every 2 minutes, is insufficient to actually maintain even just good cardiovascular health. Because I think one of the major criticisms in high intensity training is that at the time you're actually using the glycolytic pathway. So when you're using the glycolytic pathway, you're depriving the mitochondria of oxygen. And when you're depriving mitochondria of oxygen, you know that it's a hormetic stress for them, and it will induce mitochondrial biogenesis. However, if you do that all the time, then you're gonna kill your mitochondria by not giving them oxygen.
So just to be clear, you're saying 2 times a day of high intensity interval training, 7 days a week?
10 minutes.
Okay, 7 days a week or some rest days?
For me, I just recommend 3 times a week.
3 times a week. Okay.
Yeah, 3 times a week. Yes.
So on those 3 days, you do it 2 times per day.
Yes.
Okay,
yes.
Gotcha.
It's just my recommendation. If you can't, just do it once a day. Something is better than nothing, right?
Okay.
If I see a person walking, then that's good enough. Or a really big person trying to run, I stop the person and actually say, hey, you know what? Probably you're better off just walking rather than destroying your knees. So anything, any movement is better than no movement at all, right? So but again, we're not intended to go chronic use. So, and of course, it's also practical, especially for people that they're with their families on weekends, Saturdays and Sundays. So much of this goes out the window, right? So it's more practical for them to have control of their time during the week. That's my recommendation. It's come from experience. Yeah.
Okay. And just real quick on the inflammation and NF-kappa B, kind of this inflammatory signaling cascade, and NRF2, the kind of cell defense, antioxidant defense system. Hormesis ties into that because hormesis is strengthening some of those antioxidant defense mechanisms. Is that right? So hormesis ties into in a way, it can counteract some of the inflammaging process to some extent?
Yes, because there's mitochondrial memory, right? So it's preserved in the molecules that are in the mitochondria, it's preserved in the DNA of the mitochondria, and so on. So if you have hormetically induced stress, see, the equivalent is like this. Since the mitochondria have naked DNA, they have no histones, they have nothing, right? They're circular and so on. They have to preserve their epigenome via the molecules that are floating around in them and inside themselves. So, but that's why they mutate very rapidly. So each time you induce hormetic stress, then that's where it's going to affect it. So the future mitochondria that are created will actually have that kind of memory in it. So it's more resilient to stress, right? Whereas in the big epigenetics picture of your body, it is actually stored in the chromatin material, or in the material that's outside of your DNA, and that's where all the changes are made, and that is also heritable. So we're just basically mirroring, what's happening to the somatic cell and the mitochondrial cell.
Yeah, beautiful. So the high intensity interval training exercise stuff is great. I'm wondering if you have maybe 2 or 3 more quick tips that you can give to improve mitochondrial health. And also, just as a side note to this, I'm wondering if you have any tips or tricks up your sleeve to help people who are locked in cell danger response mode. Have you found anything to be particularly effective in helping people get unstuck from that?
You mean other than Suramin? No, I'm kidding.
Other than Suramin, yeah.
Ari, it's really very cheap and very simple. A lot of the things about mitochondria can really be addressed by lifestyle, and it's the most difficult thing to address, right? So, my first advice is get adequate sleep. All right, because it's the time. Get adequate sleep.
Okay.
Because it's the time that your body is actually regenerating itself. It's producing new mitochondria for you. It's doing all sorts of repair mechanisms for you. It's preparing you for the next day. I have what I call a sleep anchoring technique, which I tell my patients. I devise this. I tell them that your day begins at the time that you sleep. This is so that you never forget that sleep is important to you. It's the first activity that you should pay attention to.
Yeah, that's beautiful. I love that.
Yeah. So we go to it in terms of lifestyle. So you go sleep. When you wake up, hydrate yourself. Your body is begging for water in the morning. It has used up a lot of that water. As an aside, we were talking earlier. There's a paper that showed that mitochondria actually produces its own deuterium depleted water for the entire cell to use in order to prevent cancers. And that's just a very new study that came out. Just hydrating, you don't need to drink deuterium depleted water, just nice clean water. In order to hydrate yourself, because you're 60 to 80% water anyway. Right? And most of you are cell membranes, and therefore most of you is fats, right? So if you take a look at it that way, then you address it immediately. And then you were all yoked to the sun. And that's what we're forgetting. We have to sun ourselves. Vitamin D is not enough. UVA has its own merits. You go in the sun. We are intended to actually go and follow the sun signals. We are what's called heliotropic organisms. Right?
Yes.
We follow the sun signals. And then you make sure that you cut down on refined carbohydrates and have a lot more. The fastest disappearing macronutrient from the human diet is fiber. So we need about 34 grams of fiber daily. That not only addresses your mitochondria, it also decreases your need for carbohydrates, because your net carbs will be lower for you if you eat a lot more fiber. And as we talked about earlier, this gut microbiota actually talk to your mitochondria on a bacteria to bacteria basis. So if you're able to, if you're not able to eat well, then at least time your eating such that you're in a narrow time window enough to give your mitochondria a rest. One of the worst culprits, as I said, is ingesting too much of sugar. I was just looking at a poster lately of some heart association, showing pork chitlins as a culprit. It was morphing into a wreath. And in response, I put a sugar jelly news instead. What about this? And because the body is intended to use all these sources of fuel, we must train it to adapt to the source of fuel that it needs in order to meet its goals, right? So if you're going to be doing a lot of marathoning and you're a competitive triathlete and so on and so forth, there should be a timed way to give you your carbohydrates. There should be if you're going to be sitting on your office the whole day, of course it would be calorie restricted. People think it's ad libitum, where we could actually give you your majority of your fuel in fats. So but the thing that I'm strict about, if you want to optimize your mitochondria while managing your diet, you have to optimize your micronutrients all the time. Micronutrients are your vitamins, minerals, and cofactors that make not only the cell itself harm, but or the mitochondria itself, the cofactors that it needs to do its work. You can measure those now, and if you can afford it, go get yourself measured, and then do the proper supplementation for them.
Well, is there any particular test that you recommend for assessing nutrient deficiencies?
Yeah, right now there's a urine metabolites from Genova, for example. I use that. It's not expensive, and you don't have to draw blood. You can just take a look at urine and so on. But if you want to take a look at amino acid components, etc., then you have to draw blood. Other thing that you can do for yourself is test for your food sensitivities, because those can throw off reactive oxygen species or reactive nitrogen species, when they cause inflammation. When these cause inflammation, of course, you're increasing the molecular intermediates of inflammation. Your body has to deal with that. And then if you cannot deal with that, then your aging goes faster, right?
And then eliminate via elimination diets is your preferred.
Yes, but you know what I do, as I said, something that the patient can understand quickly. What I do is just I remove everything that they're sensitive to for 6 months, and then reintroduce it 1 month at a time, and then introduce it once every week, and then minimum distance is 1 to 4 days. It's so fucking difficult to do elimination diets. So I just say eliminate everything for 6 months. Because your gut microbiota is going to change at the time.
Yeah.
So that's what people who do elimination diets in a very strict fashion fail to understand, that the food is pre-processed by the gut bacteria, and gut bacteria is going to change dynamically every day. So there is no point in actually controlling. Just remove everything. What is easy for your patient? Do you want your patient to do something or your client to do something or not? So make him do something that he can remember.
Yeah.
Right. And then, of course, my final piece of advice is, aside from eating and exercising properly, moving well, right? Move properly, is to love well. Meaning, get rid of your toxic relationships, get rid of toxic bosses, or let your toxic bosses get rid of you. Find something where your stress is actually lower. Not that low, because you need hormetic stress, right? You need to be pushed every now and then. As I said, we're not intended to be in full parasympathetic mode all the time, and we're intended to be in full sympathetic mode all the time. Unfortunately, this world, this environment that we have created, pushes us into full sympathetic mode all the time. Like, your texts need to be answered right away. So if you develop healthy lifestyles, controlling your time, and I'm talking about relationships here, because it's your relationships to your devices. It's your relationships to your device. You have to develop healthy relationships to your devices. Not even with the people that are messaging from there, but how you deal with your devices. When do you pick them up, and when do you actually keep them away from you? So these are the kinds of things that will actually make your energy production more even, because then the brain is the brain is saying, okay, we're in good shape here, and the mitochondria say, okay, we're not being forced to produce more energy, and therefore we're not going to produce more of those reactive species that might destroy you.
Yeah. Beautiful. Well, I love that. That's such a great way to end. Love well. Perfect way to end. So, Dr. Ted, this has been an absolute pleasure and an honor to do this with you. I've been looking forward to this for a really long time, and thank you a million times over for spending all of this extra time. This is like double the length of a normal podcast episode, but there was just so much I wanted to talk to you about. So thank you for spending all this extra time with me. I really appreciate it.
You're welcome. I really wanted to do this podcast, and I know Ari, for those who don't know this, Ari has been trying to catch me since a long time, and I was in Asia, and I really owe him this time, and I think I hope I did it justice for you.
Oh, definitely. This has been amazing. So on a final note, you speaking of all your travel, you have a tri-continental medical practice. And,
Yes.
for everyone listening who hopefully is amazed with everything that you just explained on this podcast, who might be interested in working with you in your anti-aging nutritional medicine practice, how can people reach you and get in contact with you?
I started a nonprofit here in the US called healthoptimizationmedicine.org. And you can go look us up. It is just a simple website right now, and all of the physicians and health practitioners, non-physician health practitioners who are interested in practicing the way I do, which is detecting and correcting subtle toxicities and borderline deficiencies in the metabolome. That's where we all go. So you can reach me from there. You can also reach in Asia. I'm there 30 days every quarter. You can reach me at biobalanceinstitute.com. And that's based in the Philippines. And very happy to actually announce here that the first comprehensive metabolomics laboratory in Southeast Asia, which I pushed for building 4 years ago, is now going commercial this June.
Oh,
so,
awesome.
Yes. So now more problems for physicians who don't like to learn their Krebs cycle all over again.
Well, Dr. Ted, this, like I said, just been an absolute pleasure and an honor to do this with you. Thank you so much for taking all this time to do this. And I know my audience is going to love this. Thank you. And have a wonderful rest of your day.